Welcome back everyone. Today we are going to discuss this evergreen topic of glycolysis. Okay, glycolysis. Let's get started with it. So this glycolysis. Okay, first this is coming from Greek words. Okay, first is glycis, which means sugar, and lysis, which means splitting. Okay, glycis, which means sugar, and lysis means splitting. So this is essentially splitting of sugar. Sugars we have in our body basically means splitting of the sugar. Okay, now it takes place in all organelles, in cytoplasm of cells. It takes place in all organs, not organelles, in all organs. Okay, in the cytoplasm of the cells. Okay, now remember, this pathway takes place in well-fed state, that is high insulin glucagon ratio, that is in the well-fed state. This glycolysis takes place. This is also called EMP pathway. Okay, what is this EMP? It stands for Mbbs Meyer-Hoff Mbbs pathway, EMP pathway, which is our glycolysis. Now before going to the steps of glycolysis, we must see its significance. Okay, significance of glycolysis. So must be very important significance. That's why it's taking place in all the organs. Okay, first is the only pathway to take place aerobically and anaerobically. The only pathway. Next is in RBC. In RBC it is the only metabolic fuel is glucose. Okay, in both fed and fasting state, only metabolic fuel for RBCs or glucose. Okay, also mitochondria is low, so low mitochondria in RBC. We know RBC lacks nucleus as well as mitochondria, so hence occurs anaerobic glycolysis. Remember, anaerobic glycolysis in RBC. So defect in any enzyme, okay, cause lysis of RBC. Defect in any of the intermediate enzymes in the pathway, we are going to see after. The pathway will cause lysis of RBC. Defect in any enzyme, anaerobic glycolysis, and glucose in both fed and fasting state in the RBC. Okay, this is the significance in RBC. Next, we are going to see in skeletal muscle. Skeletal muscle hereby have enormous capacity for glycolysis. So in an exercising muscle, there is decreased oxygen supply. Okay, decreased oxygen supply in an exercising muscle. Here anaerobic glycolysis can operate. Here anaerobic glycolysis can also operate in exercising muscle when there is decreased oxygen supply. So any defect in glycolysis, so basically defect in any enzyme on the glycolytic pathway can lead to muscle fatigue. Okay, this was for skeletal muscle. Next we are going to see in our all important cardiac muscle. So remember here, cardiac muscle has an important point: very low glycolytic capacity. Okay, so in cardiac muscle they are susceptible to ischemia. Remember, this means reduced blood supply. Reduced blood supply is ischemia. So cardiac muscle, due to very low glycolytic capacity, is susceptible to ischemia, which can prove sometimes fatal due to many conditions. Okay, the fifth significance is glycolysis provides carbon skeleton. Okay, carbon skeleton for synthesis of non-essential amino acids. For non-essential amino acids, glycolysis provides the carbon skeleton. How? We'll discuss in further. Okay, the sixth and last here is, it is the preliminary step before complete oxidation of glucose. Okay, it is a preliminary step before complete oxidation of glucose. Glycolysis is the preliminary step before complete oxidation of glucose in the TCA cycle. How it's going to occur, we'll see further down. Okay, now these are the significances of glycolysis in RBC, skeletal muscle, cardiac muscle, synthesis of non-essential amino acids. Okay, now finally we are going to see the steps. Okay, steps of glycolysis. Okay, there are two phases of step. First is the preparatory stage. Okay, it is the preparatory stage. It is the first is the stage of phosphorylation. Okay, phosphorylation takes place in this preparatory stage, and also the stage of splitting. This is the splitting stage, phosphorylation stage and splitting stage. And after preparatory there is payoff. It is release of the energy in the form of ATP. In the payoff phase there is series of oxidative phosphorylation. In the payoff phase for releasing of energy. Preparatory stage, phosphorylation and splitting stage. Okay, now first is our preparatory stage. Okay, preparatory stage. So we start with glucose. The first compound entering the cycle is glucose. Okay, glucose then converted to glucose 6-phosphate. Preparatory stage of phosphorylation, phosphate is added, glucose is converted to glucose 6-phosphate. We are going to see the enzymes and coenzymes later. First we are going to see all the intermediate products here. Okay, so this step, formation of glucose 6-phosphate, is the first irreversible step. Okay, this is the first point. Second point, this is the one of the regulatory step. Remember, one of the regulatory step, the first irreversible step, and this is also the flux generating step. Okay, this creates the gradient of the flux needed for the further reactions, formation of glucose 6-phosphate. Okay, then this is further converted to the third product, it is fructose 6-phosphate. Very simple, glucose 6-phosphate, by isomerization we form fructose 6-phosphate. Okay, then from fructose 6-phosphate we form fructose 1,6 bisphosphate. Fructose 1,6 bisphosphate. Okay, and we have seen 4 substitution products, intermediate products. Now this formation of fructose 1,6 bisphosphate, okay, remember it is the second irreversible step. Second irreversible step. Glucose 6-phosphate formation was first irreversible step. This is the second irreversible step. This is also the committed step, also called bottleneck of pathway. What do you mean by bottleneck or committed step? That there is no going back from this formation. So that is why this is also the, remember, rate limiting step of glycolysis. Rate limiting step of glycolysis, formation of fructose 1,6 bisphosphate. Okay, now this we have seen the phosphorylation part of the preparatory stage. Now there is splitting. Now comes the splitting phase in the preparatory stage. So the fructose 1,6 bisphosphate splits into, remember, dihydroxyacetone phosphate, or simply dihydroxyacetone phosphate, DHAP, and also glyceraldehyde 3-phosphate. Glyceraldehyde 3-phosphate. This is glyceraldehyde 3P. Okay, so this is all about the preparatory stage. We have seen phosphorylation and splitting stage. Now we will see the all important enzymes and their coenzymes. So glucose to glucose 6-phosphate, first enzyme, remember, hexokinase. These kinase enzymes are known to use ATP. So the first enzyme is hexokinase. So naturally 1 ATP is used here for conversion to 1 ADP. Very simple. Then from glucose 6-phosphate to fructose 6-phosphate, I have said by isomerization. So here enzyme is phosphohexose, phosphate for phosphohexose, 6-carbon isomerase, or simply you can also say isomerase enzyme. Okay, from glucose 6-phosphate to fructose 6-phosphate. Then from fructose 6-phosphate to fructose 1,6 bisphosphate, rate limiting step. So here comes the rate limiting enzyme. What is the rate limiting enzyme here? Remember, phosphate group, fructose kinase. So there are two types of phosphofructokinase, 1 and 2. Here it is 1, EFK1 is the rate limiting enzyme of the glycolysis. Okay, so here it is kinase. Naturally it will use ATP for formation of 1 ADP molecule. Okay, these two steps very important. We will use further later this consumption of ATP. Okay, now this fructose 1,6 bisphosphate is formed the bottleneck step, rate limiting step of glycolysis. Then there is no going back. Now for this splitting, okay, we have our enzyme, remember, aldolase enzyme, aldolase enzyme. Okay, this is the split glycerylate 3-phosphate and dihydroxyacetone phosphate. Now these two, remember, these two compounds are reversible. Reversible in how? In what sense? These are isomerically reversible. So here this reversible pathway is maintained by this enzyme, remember, phosphotriose isomerase. Phosphate group, triose, 3 carbon, these are 3 carbon compounds. Okay, these are 6 carbon compounds split into identical 3 carbon compounds. Triose isomerase, these are isomerically significant compounds, both of them. This was the preparatory stage involving phosphorylation and splitting. Now the payoff phase. Okay, the payoff phase. The payoff phase. Okay, we're going to see. So as these two are similar, isomerically similar compound, so we're going to use both of them as single molecule using twice. Okay, so I will use here 2 times glyceraldehyde 3-phosphate. Okay, 2 times glyceraldehyde 3-phosphate means dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. Okay, so these two combine. Okay, these two combine to form, remember, 1,3 bis phosphoglycerate. 1,3 bis phosphoglycerate. This is 1,3 BPG bis phosphoglycerate. Remember, from this arrow you must have seen this is the reversible cycle. Okay, the reversible cycle. Here inorganic phosphate is added. Okay, 1,3 bisphosphoglycerate. Okay, here we are going to see the enzymes here. So the enzymes is, remember, glyceraldehyde 3-phosphate dehydrogenase. Dehydrogenase enzymes adds inorganic phosphates to glyceraldehyde 3-phosphate. So glyceraldehyde 3-phosphate dehydrogenase is the enzyme. Here inorganic phosphate is added. Okay, here one important thing to know, here NAD plus, okay, as this is the payoff phase, NAD plus is converted to NADH plus H plus, NADH plus H plus, and getting back into NAD plus. Okay, this is occurring in the regulatic phase. Okay, in the ETC cycle. So here remember, 1 NADH produces in ETC 2.5 ATPs. Here 2 glyceraldehyde 3-phosphate are getting converted, so 2 NADH are used. 2 NADH produces in ETC how much? Very simple, 5 ATPs. So first time ATP is produced rather than used in this payoff phase. Here NAD plus is converted to NADH plus H plus, which is getting converted to back in here electron transport chain. Okay, which therefore produces 5 ATPs in return. Okay, now in the next steps, this 1,3 bisphosphoglycerate getting converted to 3 phosphoglycerate. Okay, 1,3 bisphosphoglycerate is converted to 3-phosphoglycerate. Here remember the enzyme is 1,3 bisphosphoglycerate kinase enzyme. Okay, this is, remember, the only reversible kinase. As this is a reversible kinase, we've seen before, kinase enzymes uses ATP. Here instead 1 ADP will be converted to ATP. Okay, not just 1 ATP, here 2 ATPs will be formed. Why? Because 1,3 bisphosphoglycerate is converted to 3-phosphoglycerate, so 1 phosphate is getting removed here. So for 1 phosphate, this 2 ADP will be converted to 2 ATPs. Okay, so this is the funda. Remember, this is the only reversible kinase, which is 1,3 bisphosphoglycerate kinase. Remember, this is the substrate level phosphorylation. This formation of 3-phosphoglycerate. Now from 3-phosphoglycerate, next we are getting what? 2-phosphoglycerate. Now these compounds are not much important, just to remember the namesake. Here remember the enzyme is 3-phosphoglycerate mutase. Okay, mutase enzyme. From 3 position to 2 position, the phosphate group is shifted, 3-phosphoglycerate to 2-phosphoglycerate. 3-phosphoglycerate mutase in the enzyme, the kinase enzyme, here the mutase enzyme. Now from this 2-phosphoglycerate, in the next step, water is removed. Okay, the enzyme, remember, here is enolase. Very important enzyme. We'll see further how it is very important. This enolase enzyme requires magnesium or manganese ions, Mg2 plus or Mn2 plus required by enolase. Upon removal of water, there is formed our phosphoenol pyruvate. Is our PEP. Phosphoenol pyruvate is formed. Removal of water by the action of enolase enzyme. Okay. Now here, by the action of, remember, on the PEP, pyruvate kinase enzyme action, very important enzyme. Pyruvate enzyme action. PEP is finally converted to pyruvate. Pure pyruvate, phosphoenol pyruvate to pyruvate. Phosphate group is removed, so here ADP is converted to 2 times ATP. So here there is production of 2 ATPs. Okay, 2 ATPs produced. Here 2 ATPs produced. Here 2 ATPs are produced, and here 5 ATPs are produced. Where? In this step. Okay, we'll see across later in the energetics how they are getting fused together. Okay, now this formation of pyruvate is, remember, this step is substrate level phosphorylation. And this is the irreversible step. This is one of the, remember, regulatory step. Okay, so upon this formation of pyruvate, till this formation of pyruvate, we've seen all the 9 steps. Then 10th step, the formation of 10th product, we see the anaerobic glycolysis. Okay, anaerobic glycolysis. Here our pyruvate is converted to lactate by the enzyme lactate dehydrogenase, very important enzyme. Here one NADH is being converted to NAD plus. Okay, so here there is no net generation of NADH. NADH is getting used, so there is no net generation of NADH. So this is last product of our anaerobic glycolysis. So this all concludes our total of glycolytic pathway. There is this phosphorylation phase, use of ATP and ADP, and splitting phase, and there is payoff phase, production of ATPs, substitutable phosphorylations, formation of phosphoryl pyruvate, and finally formation of lactate from pyruvate. So formation of lactate from pyruvate, the ultimate end product of glycolysis. Okay, now we're going to see how the ATPs that are used or produced are getting fused in this segment of energetics. So how many energy are produced from one glucose or one fructose that is entering into glycolytic pathway? Okay, so first we are going to see aerobic glycolysis. Okay, aerobic glycolysis, we are going to see the first is number of ATPs produced. Okay, we have seen from conversion of 2,2-glyceraldehyde 3-phosphate to 1,3 bisphosphoglycerate. How many ATPs? 5 ATPs are produced. Okay, you can go back and see. Then in the production of from 1,3 bisphosphoglycerate to 3-phosphoglycerate. Okay, here 2 ATPs are produced. First 5 ATPs, then 2 ATPs. Then in the penultimate state from PEP, phosphoryl pyruvate, to pyruvate through pyruvate kinase, here also 2 ATPs are produced. So total ATP produced equal to 9 ATPs. 5 plus 2 plus 2, 9 ATPs. Not only ATPs are produced, also ATPs are used. Where? In the preparatory stage. Number of ATPs used. Preparatory stage, the first step itself, from glucose to glucose 6-phosphate or hexokinase enzyme, 1 ATP is used. Okay, then in the third step, that is a rate limiting step, fructose 6-phosphate to fructose 1,6 bisphosphate, phosphofructokinase 1 enzyme, 1 ATP is used. Total 2 ATPs are used. Okay, so net gain of ATPs, 9 are produced, 2 are used, which is equal to 7 ATPs. So this is the total net gain of ATPs. 7 ATPs are total produced from one glucose. What is this one glucose? 6 carbon. 1 glucose 6 carbon, 7 ATPs are produced from one glycolytic cycle. Where? In aerobic glycolysis. Okay, now in anaerobic glycolysis, anaerobic glycolysis. Okay, remember in anaerobic glycolysis one little difference is there, that this 2 into glyceraldehyde 3-phosphate to 1,3 bisphosphoglycerate. The enzyme is glyceraldehyde 3-phosphate dehydrogenase. This step does not occur. Okay, this step does not occur in anaerobic glycolysis. Okay, so as this step does not occur, number of ATPs reduced. The rest of the two steps occur. It is 2 plus 2 is equal to 4 ATPs. Now in the payoff phase, or in the preparatory phase, there are number of ATPs used. Those two steps remains the same. It is 1 plus 1 is equal to 2 ATPs are used. Okay, so net gain of ATPs in anaerobic glycolysis. Anaerobic glycolysis 4 minus 2 is equal to 2 ATPs. In aerobic glycolysis 7 ATPs. In anaerobic glycolysis this step does not occur, so no more 5 ATPs are produced. So 4 ATPs are total produced, 2 ATPs are used in the preparatory phase, so net gain is 2 ATPs. Okay, so this was the glycolytic pathway. Now we are going to see total the whole process of from 1 glucose, okay, by aerobic oxidation. So this aerobic oxidation involves glycolysis, the pyruvate dehydrogenase molecules, and the TCA cycle. Okay, so first is the glycolysis. Okay, 7 ATPs. Okay, now remember pyruvate dehydrogenase PDH complex. This glucose enters, produces, remember, 2.5 into 2, which is our 5 ATPs. 5 ATPs from pyruvate dehydrogenase complex. And finally, in the TCA cycle, it occurs twice, which is from 1 glucose 10 ATPs. So twice, which is equal to 20 ATPs. So total, if we add total, which is 7 plus 5, 12 plus 20, 32 ATPs from 1 glucose 6 carbon by aerobic oxidation. So the difference between aerobic oxidation and glycolysis. Okay, aerobic oxidation involves pyruvate dehydrogenase complex from which 5 ATPs are produced, and then finally from TCA cycle 20 ATPs are produced. So total 32 ATPs from 1 glucose by aerobic oxidation. So this was glycolysis, the energetics, their enzymes. Okay, from where the energies are produced, from where energies are used, payoff phase, the preparatory phase, the significance of glycolysis. Okay, so this is a very important topic. This is the first part of our glycolysis discussion. Do revise this topic, guys. I'll see you in the part 2 of this glycolysis discussion. Thank you. Welcome back everyone, guys. Today we are going to continue our discussion on glycolysis in the glycolysis part 2. We are going to see some enzymes, some other different cycles, some enzyme inhibitors, and also the applications of glycolysis. Okay, so we know the rate limiting enzyme is phosphofructokinase 1. Okay, phosphofructokinase 1 is the rate limiting enzyme of glycolysis. Remember, this is the allosteric enzyme. allosteric enzyme. Okay, now what is the allosteric activator? Okay, allosteric activator. Now the allosteric activator here is, remember, fructose 2,6 bisphosphate. Okay, fructose 2,6 bisphosphate is the allosteric activator of phosphofructokinase 1, which is produced from phosphofructokinase. Here it is 2. Okay, this converts fructose 6 phosphate to fructose 2,6 bisphosphate. Okay, phosphofructokinase 2 converts fructose 6 phosphate to fructose 2,6 bisphosphate. This here is the allosteric activator of phosphofructokinase 1. Okay, the right is the phosphofructokinase 1 and here is phosphofructokinase 2. So this is an inducible enzyme. This is an inducible enzyme. Okay, so the first discussion on the phosphofructokinase 1. Next, see significance of anaerobic glycolysis in RBCs. Significance of anaerobic glycolysis in RBC. Okay, so here we have our typical RBC, typical biconcave RBC. Okay, here glucose enters, converted to, remember, glucose 3-phosphate. Okay, now this is involved in NAD plus to NADH conversion, and here it is reversible reaction. Okay, we know there is no mitochondria in mature RBC. There is no mitochondria in mature RBC, so there is no electron transport chain. There is no mitochondria, so there is no electron transport chain. So NAD plus can't be regenerated. Okay, so here NADH should not be regenerated, but in RBC this enzyme, remember, lactate dehydrogenase, regenerates NAD plus, so that, remember, it does not, the lactate dehydrogenase regenerates NAD plus so that it does not accumulate in RBC. Okay, this lactate dehydrogenase is converting lactate from pyruvate, responsible for this regeneration of NAD plus, so that NADH does not accumulate in RBC. So this was the significance of anaerobic glycolysis in RBC. Remember, here glucose 3-phosphate is formed from glucose that enters into it. Okay, now I'm going to see some inhibitors of glycolysis, specifically inhibitors of the enzymes. Okay, so first inhibitor here we have in this step of conversion of glyceraldehyde 3-phosphate, obviously twice to 1,3 bisphosphoglycerate. Here the enzyme is glyceraldehyde 3-phosphate dehydrogenase, a dehydrogenase enzyme. Okay, here this enzyme, okay, first inhibited by, remember, arsenate inhibits this cross reverse inhibition. Arsenate inhibits this step and this enzyme, so this 1,3 BPG is not found upon presence of arsenate. Instead, when arsenate is present, 1 arsino 3 phosphoglycerate is formed. Okay, 1 arsino 3 phosphoglycerate is formed. So here in the next step we know this is getting converted to 3 phosphoglycerate. Okay, so here the enzyme required was 1,3 this phosphoglycerate kinase, 1,3 bis phosphoglycerate kinase enzyme. So this step does not occur. Okay, this step doesn't occur with the help of this enzyme. This step occurs spontaneously occurs. As there is spontaneous occurrence of this step, so no ATP is generated. No ATP is generated in this process. Remember, here 5 ATP was produced, here 2 ATP was produced. This does not occur. No ATP is produced due to arsenate. Only remember, arsenate is responsible for no generation of ATP. Okay. Now from 3PG, 3 phosphoglycerate, we have 2 phosphoglycerate. Okay, 2 phosphoglycerate, we have our phosphoenol pyruvate. Okay, here one important inhibitor comes in the form of, so first here is the enzyme is enolase. Okay, enolase I have said. Remember this important enzyme. Now I want to see why this is important. This enolase enzyme is inhibited by fluoride. Remember fluoride. This fluoride restricts. Remember what this enolase requires? Magnesium and manganese ions. This fluoride restricts the level of magnesium or manganese. That's why, as enolase is inhibited by fluoride, sodium fluoride oxalate. Sodium fluoride oxalate. This compound, must be familiar with this compound, used in blood banks for storage of blood. The sodium fluoride oxalate mixture used in collection tube for estimation of blood glucose. Estimation of blood glucose. Okay, it inhibits glycolysis. Here in this point, by inhibiting enolase enzyme, by restricting the level of magnesium and manganese, what? Fluoride, so that this glycolysis step does not occur and glucose remains as it is in the blood collection tube for estimation of blood glucose. So these are the two important inhibitors. First one is arsenate, for which no ATP is produced, and second is sodium fluoride oxalate, most important one, inhibits the enolase enzyme. Okay, yes. Now we are going to see some metabolic defects. Okay, metabolic defects. Metabolic defects due to enzymes of glycolysis. Okay, first is, remember, pyruvate kinase defect. Pyruvate kinase defect. I once asked in the glycolytic pathway that, remember this penultimate enzyme pyruvate kinase. Now this defect is the second most common enzyme defect in humans. This is the second most common enzyme defect in humans. Okay, here RBC is going to be affected the most. Okay, due to there is decreased ATP production and ion channels in the RBC are not maintained. Non-maintenance of ion channels and decreased ATP production, RBCs are affected the most. So there occurs calcium influx, calcium moves inside, and potassium efflux, potassium moves outside. There occurs swelling of RBCs leading to hemolysis, and we know this will lead to hemolytic anemia and other factors such as jaundice in the later studies, but hemolytic anemia will occur due to swelling of the RBCs and lysis due to pyruvate kinase defect. Very, very important. So to compensate this, to prevent from this happening, to compensate this, the body does what? This glucose rapidly converted to 2,3 bisphosphoglycerate. But this is found in this cycle Rappaport. Sorry, I'm going to write this again. 2,3 bisphosphoglycerate is found in Rappaport Lubering cycle. Okay, the glucose is converted to 2,3-BPG. Okay, which we know from physiology, this leads to unloading of oxygen. This promotes unloading of oxygen at the tissues where this is required for metabolism. There occurs unloading of oxygen, okay, due to formation of 2,3-BPG by Rappaport Lubering cycle. Okay, then next is our aldolase enzyme. The enzyme involved in splitting of fructose 2,6-bisphosphate to dihydroxyacetone and glycerol 3-phosphate, the aldolase A enzyme defect. Okay, this will also lead to, remember, hemolysis. Okay, now there is muscle phosphofructokinase defect. This will lead to, as muscle is involved, obviously exercise intolerance. Muscles won't tolerate the exercise. There will be lot of fatigue. Muscle PFK defect, exercise intolerance. Aldolase A defect, hemolysis. Okay. Now is another last one to discuss. This is maturity onset of diabetes in young. This is MUDI, MUDI 2, which is 2, maturity onset of diabetes in young, which is 2. This is due to the kinetic property of glucokinase. What is this glucokinase? Isoform of hexokinase. Okay, kinetic property of glucokinase, which is the isoform of hexokinase, is altered. Okay, this kinetic property of glucokinase is altered, then which will lead to the ATP buildup in pancreatic cell, where insulin is produced. ATP buildup in pancreatic cells is affected. Not enough ATP is produced, so insulin can't be secreted even in hypoglycemic conditions. Even in hypoglycemic conditions, this insulin can't be secreted due to the ATP buildup in the pancreatic cell being affected. Why? The kinetic properties of glucokinase, which is an isoform of hexokinase, being altered. So this will lead to maturity of onset of diabetes in the young. MUD by 2. Okay, this is onset. So this is O. Okay, MOD by 2. So these are all the enzymatic defects leading to various serious diseases in humans. Okay, now we are going to see some regulation. Finally, we are coming to regulation of glycolysis. Okay, regulation of glycolysis. Okay, yes, we are going to see some enzymes. Okay, there is allosteric activator of the enzyme. Okay, and there is allosteric inhibitor of the enzyme. Allosteric activator and allosteric inhibitor of the enzyme. Okay. First important enzyme you have to see is hexokinase. First enzyme in the glycolysis, hexokinase. There is no allosteric activator, but the allosteric inhibitor here is the ATP or bracket phosphate. You can remember the product ATP always inhibits the reaction. It tries to push the reaction backwards, so ATP will inhibit the hexokinase enzyme. This is the allosteric inhibitor. Okay. Next enzyme, rate limiting enzyme, phosphofructokinase 1. Okay, there are allosteric activators we have discussed here. And the starting of this discussion is fructose 2,6 bisphosphate. Okay, also fructose 6-phosphate, fructose 6-phosphate and fructose 2,6 bisphosphate, along with, remember, 5-AMP. So fructose 2,6 bisphosphate, fructose 6-phosphate along with 5-AMP are allosteric activators of phosphofructokinase 1. Now the allosteric inhibitors is the most commonly asked question. Also here, as ATP is produced, ATP, okay, the form of phosphate. Also remember citrate, very important, most frequently asked, citrate. And then there is low pH. So low pH, citrate and ATP are the allosteric inhibitors, inhibitors of phosphofructokinase 1. 5-AMP, 6-phosphate and 2,6 bisphosphate of fructose are activators, allosteric activators of PFK1. And finally, we have pyruvate kinase 1, or simply pyruvate kinase. We don't have any allosteric activator here, but here also the allosteric inhibitor is ATP. Okay, allosteric inhibitor is ATP. Okay, the ATP is almost everywhere the allosteric inhibitor. To remember for this, especially the allosteric activators and inhibitors. Okay, pyruvate kinase and hexokinase inhibitors are ATP, allosteric inhibitors. Okay, this was all about our second discussion on the glycolysis. Okay, I've seen the enzyme defects relates to what? Some inhibitors of the glycolytic enzymes, the significance of anaerobic glycolysis and RBCs, the rate limiting enzyme, their allosteric activators and inhibitors, important enzymes, activators and inhibitors here. Very important discussion. So please keep revising the 2 modules. Thanks for watching. I will catch you in the next one. Good luck.